Hello, my dear students. Welcome back to the channel. And today we are going to discuss a topic glycolysis. That topic covers the concept of respiration. And in this topic, we are going to discuss how the glucose respiratory metabolism starts. This topic covers A-level biology and it's equally applicable in Cambridge, Edexcel, OCR, and AQA. Glycolysis is the base process of respiration, whether it is aerobic respiration or it is anaerobic respiration. Glycolysis is the base process. Glycolysis refers splitting glucose molecule, that is a 6-carbon molecule, into 2 molecules of 3-carbon sugars. Why it is done? It is actually to increase energy efficiency from 6-carbon glucose molecule. Glucose is the primary respiratory substrate that is metabolized in the respiratory pathway and energy is yielded mainly in two forms, primarily in the form of ATP and in the form of heat. During glycolysis, glucose molecule needs to be split into 2 molecules of 3-carbon sugars. But glucose molecule itself is inert molecule, least reactive. And in order to split it, it needs to be activated, energized. And this is done with the help of an ATP molecule. An ATP molecule in the beginning of the glycolysis, an ATP molecule is hydrolyzed and phosphate and energy released is added with the glucose and it is converted into glucose 6-phosphate. Remember one thing, this process is the phosphorylation of glucose, and addition of phosphate to any molecule increases its energy level. Now the glucose 6-phosphate is energized but is not sufficiently energized that it can split into 2 3-carbon molecules. So it needs to be further energized. But for further energizing, it needs another phosphate input. But it does not get another phosphate. Why? Because its geometrical shape, or you can say its 3D shape, does not allow it to accept another phosphate. So it is isomerized into fructose 6-phosphate, and fructose 6-phosphate is able to accept another phosphate. Now in this third step, another phosphate is actually obtained from the hydrolysis of ATP along with the energy, and this phosphate, when added to fructose 6-phosphate, it is converted into fructose 1,6-bisphosphate. Now this 6-carbon molecule with 2-phosphate is sufficiently energized that it can no longer stay as fructose 1,6-bisphosphate. It immediately splits into 2 molecules of 3-carbon sugar. If you people are taking exams through any of the boards, Cambridge, Edexcel, AQA or OCR, and you are taking biology exam, please subscribe this channel and contact me for quality education, for quality guidance to achieve maximum possible grades in your exam. One is dihydroxyacetone phosphate and the other is glyceraldehyde 3-phosphate. Dihydroxyacetone phosphate is immediately converted into glyceraldehyde 3-phosphate, and now we have 2 molecules of glyceraldehyde 3-phosphate. Each glyceraldehyde 3-phosphate is a 3-carbon molecule with 1 phosphate. In the next step, hydrogen is released, and this process is known as oxidation. Hydrogen is released from each molecule of glyceraldehyde 3-phosphate, and that hydrogen is replaced with inorganic phosphate from the cytoplasm. So this process is known as oxidation because it's the removal of hydrogen. And removal of hydrogen is only possible with the help of a hydrogen carrier. Over here we have got a hydrogen carrier known as NAD. This is named as nicotinamide adenine dinucleotide. This nicotinamide adenine dinucleotide, abbreviated as NAD, in its oxidized state, it accepts hydrogen that is released from the oxidation of glyceraldehyde 3-phosphate. So 2 hydrogens are released, 1 from each molecule, and as a result, after accepting these 2 hydrogens, we have 2 reduced NAD molecules. Now the space generated after the loss of hydrogen is captured by the inorganic phosphate. So inorganic phosphate ions that are present in the cytoplasm, these inorganic phosphate ions are added up, one in each molecule of glyceraldehyde 3-phosphate. So after oxidation and addition of inorganic phosphates, so these two molecules are converted into 3-phosphoglycerates. And this is actually 3-phosphoglycerates, not, but actually it's 1,3-bisphosphoglycerates. Means each 1,3-bisphosphoglycerate is 3-carbon molecule with 2 phosphate added. In the next step, this 1,3-bisphosphoglycerate is actually dephosphorylated, and phosphate is removed from each molecule. So we have got 2 molecules of 1,3-bisphosphoglycerate. So 1 phosphate from each molecule is removed, and that phosphate, along with energy that is also released along, is taken up by the ADP molecules, and they are converted to ATP. Now the 2 phosphates are released and 2 ATP molecules are generated at this stage. Over here, energy is derived from the conversion of 1,3-bisphosphoglycerate into 3-phosphoglycerate molecules. So this is known as substrate level phosphorylation. In the next stage, 3-phosphoglycerate is converted into 2-phosphoglycerate. In the very next stage, 2-phosphoglycerate molecules lose water, 1 water from each molecule, and it is converted into phosphoenol pyruvate. At this stage, we have got 2 molecules of phosphoenol pyruvates, and each phosphoenol pyruvate has 3 carbon and 1 phosphate. In the very last stage of this glycolysis, from each molecule of phosphoenol pyruvates, last phosphate is removed and it is converted into phosphate. It is converted into pyruvate. The phosphate that is released from each molecule of phosphoenol pyruvate is taken up by ADP and it is converted to ATP molecule. Again, this is substrate level phosphorylation. Now, at the end of glycolysis, what we have, if you people are taking exams through any of the boards, Cambridge, Edexcel, AQA or OCR, and you are taking biology exam, please subscribe this channel and contact me for quality education, for quality guidance to achieve maximum possible grades in your exam. We have 2 molecules of pyruvate. Each pyruvate is a 3-carbon sugar. But in the last stages of glycolysis, we obtain 4 ATP molecules. 2 from the step where 1,3-bisphosphoglycerate was dephosphorylated into 3-phosphoglycerate, and 2 molecules of ATP were obtained when phosphoenol pyruvate molecules were converted to pyruvate. So 4 ATP molecules were obtained at the end of glycolysis. Remember one thing, that during this glycolysis process, energy efficiency is very low. Hardly there is a net gain of 2 ATP molecule, and each ATP molecule contains 30.5 kilojoule energy, or if we round it off, so 31 kilojoule of energy. So the total energy gained, total net energy gained by 2 ATP molecules is just you can say it is around 62 kilojoule. But reduced NAD molecules, the 2 reduced NAD molecules produced during oxidation process of glyceraldehyde 3-phosphate, they will be oxidized at electron transport chain on the cristae of mitochondria during oxidative phosphorylation and yielding maximum of energy in the form of ATP and also in the form of heat. In this process, the net gain what we have, when the glycolysis process started with 1 glucose molecule, what do we have at the end of glycolysis? That we have 2 molecules of ATP consumed in the beginning, 4 molecules of ATP produced at the end of glycolysis. And the net gain of ATP molecule is 2 ATP molecules. We obtain 2 reduced NAD molecule. They are likely to be oxidized during oxidative phosphorylation at electron transport chains on the cristae of mitochondria. And 2 molecules of 3 carbon sugars known as pyruvates. Now the fate of the pyruvate will be decided whether the aerobic respiration needs to proceed, and in case of aerobic respiration, these pyruvate molecules will be transported to the mitochondria and they will be further processed into link reaction, also known as pyruvate oxidation. And the products of pyruvate oxidation will be processed during Krebs cycle. And if there is anaerobic respiration going on, so pyruvate will be further processed, oxidized within the cytoplasm, and that could be lactate fermentation in case of muscle cells, or that could be alcoholic fermentation if it is yeast. And the details of anaerobic respiration are further details of aerobic pathways like link reaction, Krebs cycle, or even oxidative phosphorylation will be discussed in the upcoming lectures. So stay tuned for the upcoming lectures. Thank you very much.